hitting record so that we can look at this later. So this is for chapter 12 for antibacterial, antimicrobial therapies. This first slide here I wanted to show to you guys because these are the characteristics of the perfect or the ideal antimicrobial drug, and all of these conditions are not ever met at the same time, but they're all very important. Some of the ones that I wanted to highlight, particularly this one, is toxic to the microbe but non-toxic to the host. That's basically the definition of selective toxicity. We would like it to be cidal rather than static. It kills rather than inhibits, remains potent long enough to act, and of course, it would be best if it was reasonably priced. That goes down as the cost goes down the longer the drug is actually on the market. These next two slides, these are definitions that you must know for the exam, and they are bulleted out just like this in your textbook. The one that I want to focus on in particular, notice antimicrobials. That means all-inclusive, any drug that will kill a bacteria or a set of drugs that might kill fungus or a protist. An antibiotic is a very specific definition of a substance or a compound that's produced naturally by a bacteria or fungus, not created by scientists, that can inhibit or destroy microorganisms. The second slide of these terms that you have to know would be important to focus on: narrow spectrum versus broad spectrum. And it's very intuitive. Broad spectrum kills lots of different organisms. Narrow spectrum only kills the ones that are very few organisms or of a particular type. This is a slide that shows an example of that. Let's take a look at the tetracyclines. They would be considered broad spectrum because they can be used to inhibit or kill gram-negative, gram-positive, chlamydia, rickettsias. The isoniazid, I can't say that very well, obviously. Those at narrow spectrum, they only are going to kill tuberculosis bacteria or mycobacterium. Notice this purple line doesn't go into any of the other types of bacteria. Narrow spectrum, broad spectrum. Okay, this is a picture you guys should focus on. This shows the 5 different mechanisms or the 5 different modes of actions, or notice antibacterial drugs. So the picture here is of our friend superbacteria back from chapter 4. It is important not to know the names of the drugs and which ones do which thing, but you need to know these 5 modes of action. So the next slide, I've highlighted those 5 modes of action. They're the same as here: inhibitors of cell wall, inhibitors or changing cell membrane permeability, inhibitors of nucleic acid, inhibitors of metabolism, and inhibitors of protein synthesis or translation. Same, right? What you guys need to do in this section, I don't have time in this video to go through all of the possibilities, but you need to, I kind of highlighted here in number 1, write down as many specifics that you can remember about bacterial cell walls. And what I mean by that is they're made of peptidoglycan. What is peptidoglycan made out of? How is it assembled? And any drug that inhibits any of those parts, that would be a cell wall synthesis inhibitor. Think about inhibitors of nucleic acid synthesis. That's why I wrote this. All the enzymes involved in replication or transcription. So you guys should take a few minutes here, look in your notes, think back to chapter 9, and think about any drug might inhibit helicase, gyrase, DNA polymerase, ligase, or RNA polymerase in case of transcription. This is included in the PowerPoint set here because these are the 5 different ways that antibacterial drugs in particular can inhibit translation. And what I want you to notice is these 5 things relate specifically to pretty much the 5 steps that we talked about in translation. So for example, this first one here, this set of drugs, they inhibit the large subunit from binding to the small subunit. Okay, that would have been in part of step 1 for us when we had our steps of translation. This one, the aminoglycosides, they prevent the messenger RNA from laying down correctly in the small subunit. This group blocks peptide bond formation. This group, the tetracyclines belong to this group, they prevent the A site from being filled. And this last one, erythromycin, again, not important that you know the name, but this set of drugs prevents the ribosome from translocating or moving over, so we can never get a new empty A site. For antifungals, there's the categories are fewer. There are only 4 different major drug groups, and I would like to point out to you that's for 2 reasons. Mainly because you are a eukaryote, fungus are eukaryotes. So it's harder to find some selective toxicity because fungus and us, fungi and us, use many of the same cellular mechanisms, meaning we do replication the same, we do transcription almost the same, we do translation almost the same, we do glycolysis almost the same. So it's harder to find a selectively toxic target. I would point out the azoles. These are the ones that most of us have used over the counter for a yeast infection, for example, or for athlete's foot. And some of these can be used systemically as well. And what they mean by that is you can get them as an IV infusion, or they can be administered orally. This one in particular, I wanted you to take a look at echinocandins. That this part of the word chino, that's abbreviation or part of the word of chitin. So notice that inhibits cell wall synthesis, whereas the amphotericins or the macrolide polyenes, which are the oldest known antifungal agents, bind to fungal membranes, okay? So those 4 different groups are important for you to be able to distinguish between. The antiprotozoan and antihelminthic drugs, you'll notice that group is a lot smaller than the groups that we've talked about before, especially smaller than the antibacterial groups. And again, protists and helminths, those are both eukaryotic cell types. So again, it's difficult to find selectively toxic targets for those. You might recognize some of these drugs down here in the antihelminthic category, the praziquantel and ivermectin. Those are both given sometimes to your pets when they're, especially puppies, when they have a roundworm infection or a tapeworm infection, right? Those are both helminths. Oh, last thing I want to say about those 2 is those 2 in particular, I believe, prevent microtubule formation, so the cells can't continue mitosis. Actually, I think that's this one, the first one, and then these 2 also inhibit, they cause paralysis of the worm, and so then the worm can't digest your food. For the antiviral drugs, they also come in 3 categories. We have ones that block the entry of the virus. We have drugs that block how the virus makes a copy of its viral genome. And the 3rd category is how the virus assembles and releases. What I want you to notice about the antiviral drugs in particular is they tend to be more specific to a particular virus, meaning we can't use them for every virus. We can only use them for the virus that they're designed for. Let's take a look at some of the drugs that inhibit viral entry. So remember back to chapter 6 about viruses. Viruses need to bind or adsorb, then they undergo endocytosis, and then uncoating. So this is to block the adsorption process or the binding to the host receptors. Different viruses use different host receptors on different cells. So in this case, we've got a drug that's going to block the receptors that the HIV virus uses. And then here's one that's specific for influenza or flu virus. Clearly, those are 2 different diseases. The drugs that block nucleic acid synthesis, notice there's a little bit more variety here. We have a group that inhibits herpes virus. We have a group for RSV and some hemorrhagic fever viruses. That means Ebola and Marburg and Lassa fever. And then, of course, we have a larger collection that inhibit HIV. The reason, again, that these are specific to a particular kind of virus, think back to chapter 6. We have viruses that come in as double-stranded DNA. We have viruses that come in as single-stranded DNA. We have viruses that have double-stranded RNA for their genome, viruses that have single-stranded RNA, and that can be a sense strand or an antisense strand. And then, of course, we have multiple strands of RNA viruses. So all of those different genomes would require drugs that can prevent that kind of genome from actually being replicated. So in the case of acyclovir here that works on herpes virus, herpes is a double-stranded DNA virus. So it's going to cause termination during replication, I believe, for DNA polymerase. A virus that comes in as RNA, that's going to have no effect on it because it doesn't have double-stranded DNA. And then lastly, antiviral drugs that can prevent the assembly and release of a virus. And in this case, there's pretty much only one that's available, 2 exact drugs, but they're different versions of the same thing. And you might remember that when we looked at virus release, HIV is a virus that releases by budding. And the virus actually produces these long protein molecules. And those protein molecules need to be cut into pieces to become part of the proteins on the surface of the HIV virus, as well as part of the capsid. So it's a little bit tricky. So a protease is an enzyme that can cut protein. So the drugs are protease inhibitors. They prevent the virus proteases from cutting the long proteins into pieces so the virus can't assemble and release. When a person takes a cocktail to prevent HIV infection or to control HIV infection, they would be taking this drug, some of the drugs from this category, and then a protease inhibitor, right? Because they're going to work on 3 fronts: trying to prevent virus from binding to host cells, trying to prevent the virus from replicating, making copies of the viral genome, and then thirdly, trying to prevent the virus from being able to assemble and release to infect new cells. Okay, this section has to do with testing. It's called sensitivity testing to determine particularly if an antibacterial drug would be a good drug to use in a patient with a particular infection. So first, let's take a look over here, see how that you can see this lawn of bacteria that would have been placed on there by a lab technician from a patient's wound, or maybe if it's a urinary tract infection, by spreading the bacteria from the urine all over the Petri dish. And then these little tiny filter paper discs that have already been infused with a particular antibiotic at a single concentration, those are equally spaced around the Petri dish. Put it in the incubator, come back tomorrow, and you can notice that some of the drugs, there's this area of clearing around them, and some there is not. For every drug that has an area of clearing around it, that has a special name. It's called the zone of inhibition. It's marked here on the diagram, and that needs to be measured with a ruler in millimeters, not centimeters, in millimeters, and the whole diameter of that zone of clearing. And then the technician would consult this table. Okay, so that test is called the Kirby-Bauer test. And for example, let's take a look at the second line of the table here where they're talking about gentamicin. So if we put gentamicin on that in one of those little round discs on that Petri dish that had a patient sample on it, we would be measuring in millimeters. And as long as that zone of inhibition, the diameter of that was greater than 13 millimeters, which in this case it is 16, we would call that bacteria is sensitive to that antibiotic. Look, it is not sensitive to neomycin, right? Or penicillin G, for example. So for penicillin G, if we needed a bacteria to be considered sensitive, it has to be greater than 29 millimeters. So it's almost 3 centimeters. Notice in this example result, the Staph aureus only had a zone of inhibition of 10 millimeters, so that one would be considered resistant. Then the physician can look at those results and make a decision which drug might be the best for this particular patient. Now, there are some disadvantages of the Kirby-Bauer sensitivity test. Let's talk about the advantages first. The advantages are it is simple to do, it is relatively inexpensive, and it doesn't take a very long time. Okay, we can set this up one day and we can come back and look at the results the next day. So it's quick and fast. The disadvantages, though, sometimes outweigh that. One disadvantage is you must have the chart, right? You must have this chart Otherwise, you can't read the results. These zones of inhibition do not distinguish between bacteriostatic or inhibitory and bactericidal. And the third thing is you're only testing one concentration of the drug at a time, right? So let's say this was the best drug, but that might be too high of a concentration for a child or for an elderly person. So one way to address that, there's been an advance to the Kirby-Bauer, and that's using these E-strips, or they're called E-test strips. And they're flexible, almost plastic-like, and they have different amounts, different quantities of antibiotic in different areas of the test strip. And if you look really carefully, or especially if you take a look in your textbook, you can see that the zone of inhibition gets larger here at the top than it was at the bottom. This gives you a little bit more of an indication of what dose you might want to use to treat that particular person. The gold standard is this test. It's called the broth dilution test, and it determines the minimum inhibitory concentration. This panel is showing you two ways to do this test, two different protocols. This protocol has been done in broth tubes, and we have bacteria is added, same size of inoculum to all of the test tubes, and then we change the amount of antibiotic and we look for growth or no growth. This would be measured, the amount of growth would be measured on a spectrophotometer that we talked about at the end of the previous video for chapter 7, measuring microbial growth. This test is a little bit more, gives you a little bit more information and is a little bit more useful. What I want you to notice, first of all, is any of the containers, any of the chambers here that are blue, that indicates that the bacteria is dead. There's no growth. Pink indicates growth. So these tubes, these little compartments, they're very small. They hold less than half a milliliter. And this whole plate that's pictured here is about this big. So this is a miniaturized version, and there's 96 wells on this plate. So we can test 7 different drugs. And what isn't very clear from just this short description is the drug has actually been serial diluted in these wells as we go along. Put the same amount of bacteria in all of the wells, and then we do serial dilutions of the drug. So let's take a quick look at the results here. This drug that's in the third row, okay, flucytosine, it kills at the highest concentration immediately. Great. But notice it doesn't kill at lower concentrations. So this might be a good choice, maybe not. This one, which stands for amphotericin B, notice we can use this one, it's killing the bacteria, killing, all the way out to this dilution, whatever it is, before the bacteria can start to grow again. So this one might be a better choice because we can use it at a lower concentration. So this gives us the most information. It tells us the minimum inhibitory concentration. It distinguishes bacteriostatic from bactericidal, and you can test multiple doses at the same time. Okay, this is just a slide that summarizes that. And these are some things that the physician would need to consider, of course, before starting antimicrobial therapy or antibacterial therapy. Be best, optimum, if they knew what organism it was, but that may take several days. The susceptibility of the organism or the sensitivity, you might have to do Kirby-Bauer or the broth dilution test. And then this is something that needs to be considered. What is the overall medical condition of the patient? Are they taking other drugs that might interfere? Do they have an allergy to this particular medication, even though it's the best one to use? So it's not just as simple as getting the test results back and saying, oh, that's the right drug because it kills, right? That's too simple, too simplified. Now, this is sort of the last big portion of this chapter, has to do with how does resistance develop? And I wanted to toggle, if I could, to, I'm gonna stop the sharing, to this, what I've drawn on the board here for a minute. For drug resistance, there are 3 parts to drug resistance and the development of drug resistance by the bacteria to a particular antimicrobial drug. The first has to do with the genetic changes that occur in the bacteria. The second is how those changes affect processes inside the bacterial cell. And the last part, which is almost as important as the first two, is human behaviors that sort of help encourage resistance by the bacteria. So I'm going to go through these three things using the slides. That's the PowerPoint presentation. But first, I think I'm just going to talk about these two real quickly. The genetic changes that happen in the bacteria, they're happening all the time. Spontaneous mutation, 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 mutation. Remember, every time a bacteria divides, there's about 4,000 genes. That's 4 mutations, just spontaneously. They just happen. DNA polymerase makes mistakes. Proofreading makes mistakes. If a bacteria is dividing every 20 minutes, that means every 20 minutes there's 4 mutations. Those can accumulate quite quickly. Second portion here, once a bacteria has a mutation that actually confers resistance to a particular drug, if that mutation then is located on a plasmid, horizontal gene transfer, conjugation, transformation, transduction, okay, and or transposons. So again, check back to your notes to chapter 9 and review those real quickly. Okay, I don't have time to go through, review them all for you right this minute. So now let's take a look at, there's a diagram in your book for these genetic changes. These mutations cause genetic changes of how the bacteria sort of processes and responds to the antibiotic. So let's go back to the PowerPoint and take a look at those. Okay, so real quickly here, where are we? This picture. So this belongs to number 2 on the chalkboard here. So, and I guess I'm just going to run through them very quickly. Sometimes those mutations that the bacteria has can actually generate a new enzyme, a new protein that has a new activity. In this case, it's breaking the antibiotic. And for penicillin to be active, it has to have both of these rings. There's an enzyme that's formed by bacterial mutations that can break one of those rings. And when it does, penicillin no longer is effective. In this panel, notice the drug is binding to the bacterial cell. The receptor changed. The drug can't get in. In number 3 here, the drug is getting in. So the receptor didn't change. The drug is getting in, but there is a pump that is pumping that drug out so it never reaches its target site. The binding site for the drug, and they're using a picture of a ribosome to tell you, to try to indicate to you that the ribosome can become mutated so that it still functions and the drug can't bind to the ribosome anymore and interrupt its function. And lastly, if it's a metabolic pathway that's affected, there can be an alternate pathway that gets turned on. Even though the drug is blocking the main pathway, the bacteria has mutants that accumulate that give it a different pathway to make the same product. Maybe it's amino acid, for example, and the bacteria lives. The things that humans do, so number 3 on my list here, the human behavior that contributes to resistance. This is just a short list here. Many of the antibiotics that are prescribed are for viral infections, and so that's not going to help it's not going to kill the virus, and it's going to increase the chances that the bacteria that are living inside of you are going to become resistant because they're exposed to that pressure of the antimicrobial drug. There is a lot of excess antimicrobial drugs are produced and prescribed and exported to other countries where you may not need a prescription to actually access a particular antibiotic. You may be able to self-medicate. Hospital environments, clinical environments, nursing home environments, these are environments where a particular pathogen, say Staphylococcus aureus, can be exposed to multiple drugs over and over again, and it becomes multiply drug resistant, like MRSA, for example. And then healthcare workers or equipment that's used in the hospital can be actually an agent to transmit the infection from one person to another. This is one a lot of you may not think about. There are antibiotics that are included in feed that's given to animals, such as, let's say, chickens. And then what happens is the chicken develops like salmonella that is resistant to multiple drugs. And then you as the consumer, you get that chicken, you don't cook it well enough, or the bacteria get on your cutting board or in your kitchen and you ingest them. And then you end up with an infection of salmonella that's already multiple drug resistant before you even get your first visit to the doctor. And I think the last one here, worldwide travel, is pretty much, that's easy for us to think about, especially right now. It doesn't have to be worldwide travel, it could be state to state travel. Once a bacteria has become resistant, it's traveling with or living inside a particular person. When that person moves to another place, they take their resistant bacteria with them and share them with the people that live there. So what's to be done? How can we come up with new drugs that may not promote resistance? Using bacteriophage is one idea that people have been trying to, because they're specific. Again, look back into chapter 9, both generalized and specialized transduction, and that we could use bacteriophage lytic phages that could lyse the bacteria without using an antibiotic. Continuing to search for better targets, more selectively toxic to the bacteria, more than the host. And then the use of prebiotics, probiotics, and bacterial transfers. Sometimes the bacterial transfers, your book does a discussion there for fecal transfers, people who have multiple drug-resistant strains of Clostridium difficile. So you should look that over. Here's just a short list of some of the adverse effects. We have to bear in mind that when we take a medication, there's always going to be side effects. These are some of the systems that are targeted. You'll notice it's pretty much all of your systems. One of probably the most common side effect of taking an antimicrobial drug and the ramifications that that can have. And then, of course, I listed allergy here as an adverse effect.